Okay, the word thermodynamics isn't hugely different than bioenergetics, other than the fact that thermodynamics applies not just to living organisms, but to anything, things that are non-living. So it's a little bit broader. We're going to use it, though, only applying it to living systems. So when we're talking about energy transfers and transformations, we are talking about a system. The system can be any of the levels of the biological hierarchy. You can be talking about energy transfer with one organelle, and the organelle is the system, and then everything else beyond that is the surroundings. You can be talking about a whole cell. The cell is the system, and everything else is the surroundings. You can be talking about the whole organism as the system, and everything else is the surroundings, however you want to define it. Typically in this course, we'll be talking about the individual cell as the system, and then everything else as the surroundings. Systems can be open or closed, but in biology, if we're being more specific to biology, all biological systems are open systems. A closed system cannot, all organisms and all parts of biology interact with each other beyond just the system. So everything is an open system in biology. That's the only one you need to think about as being important for biology. So I'm going to give you two fundamental laws. You need to memorize these. I mean, like flat out recite these. The reason is these are rules of the universe. At least they're the rules that we believe are correct right now. And so it's conventional wisdom, so you need to, in science. And so you would be expected to stick to these, all right? You aren't allowed to decide that they're wrong. So these are the fundamentals that we have right now. So the first law says the amount of energy in the universe is constant. And by the way, it's the amount of matter in the universe is constant. Remember, matter is one piece of it and energy is the other piece. We're talking about energy here, though, so we're going to say the amount of energy in the universe is constant. Energy cannot be created or destroyed, but it can be transferred or transformed. Transformed means changed from one form to another. So remember, the forms of energy are kinetic and potential. So if you have a type of kinetic energy like sunlight, and the energy in sunlight gets converted into chemical energy, then that would be an energy transformation, because you change it from kinetic to potential. So a transformation means from one form to another. An energy transfer would be from one object to another. So if you have, and the object could be a molecule. So if the energy in one molecule is moved into another molecule, that would be an energy transfer. It's still potential energy, chemical energy, but it's changing objects. That would be an energy transfer. Either way, all right? But that's the first law of thermodynamics. The second law of thermodynamics says that the transfer of energy, for any transfer or transformation, a little bit of energy is always lost. Not lost, it's somewhere in the universe, but it's lost from the system. And it enters the universe in an unusable form, usually heat. And that results in an increase in disorder in the universe, and we call disorder entropy. So that's a rule. What that means is every energy transfer and transformation causes the entropy of the universe to increase. So the disorder means order would be like a macromolecule has order. It has a certain construction of smaller units. A pile of monomers is disorder. So large molecules have more order than the monomers that make them. A cell has order, and a pile of chemicals has disorder. So order is the same thing as structure. And so if you have a building made of bricks, it has a certain organization, a certain structure, that's order. If the building falls down and becomes a pile of bricks, that's disorder. What we're saying is that the universe naturally moves towards an increase in disorder. If there is going to be order, that means that work, energy, remember energy, the ability to do work, Work has to be done to maintain order. So your cells and your body are constantly trying to maintain order. They're trying to build and synthesize structures because the universe is always, in a sense, trying to break down those structures. And so there's always this battle. Eventually you'll lose the battle because you will die and your body will become completely disordered. It will become completely decomposed. So for a time, your body, while you're alive, your body will try to maintain order by transferring and transforming energy. Ultimately, you're going to lose that battle. It's a little bit depressing. So here's an example of transformations. The chemical energy in an ice cream cone, all that sugar, becomes kinetic energy that you use in your body. Light energy, that's kinetic energy, is transferred and transformed really into the chemical energy of glucose by photosynthesis. So cells are constantly trying to create order and structure from less ordered materials. So that takes energy. All right. They're always in that process. They're always going to also do some breakdown reactions as well. So there's going to be some construction and deconstruction that happens inside the cell. All right. Ultimately, in the big picture, the sunlight comes in. It gets transferred and transformed many times and ultimately ends up in the unusable form of heat. And remember, heat is increasing the entropy in the universe. So what provides the energy for a cell? You'll remember this picture here. If we had an endergonic reaction, you needed energy. What does that mean? And even when we wanted to get a reaction over the activation energy barrier, where does that energy come from? That energy comes from a molecule called ATP. ATP stands for adenosine triphosphate. Adenosine triphosphate, you'll recognize, is really one of the nucleotide molecules. So you recognize the nucleotide, but this one has 3 phosphates instead of just 1 phosphate. So you'll recognize the pentose sugar part, the adenosine, which is a nitrogenous base part, and then 1 phosphate. But this one actually has 3 phosphates. So here's what happens. Adenosine triphosphate can be broken down. Broken down means one of the phosphates is broken off. And when that happens, the breaking of that bond releases energy. It releases 7.3 kilocalories per mole, I think it is. And the cell breaks down ATP if there's a reaction that needs up to 7.3 kilocalories per mole. So each ATP is like a little packet, and when it's broken, it releases some energy, and then an endergonic reaction absorbs that energy, for example. So here's the definition of ATP hydrolysis. ATP is broken by water. It is a hydrolysis reaction, and it breaks 1 phosphate off, so that phosphate becomes separated from the rest of the molecule. And at that point, we call it adenosine diphosphate, DI, instead of triphosphate, which was TRI. So this inorganic phosphate is the one that's been broken away from the main molecule of ADP. And that releases some energy. It releases this amount of energy. And it's given a negative number because you remember a breakdown reaction releases energy. So when there's a negative number in front of it, it means that's what's released from the reaction. And that is coupled, typically, there's an endergonic reaction waiting in the wings, ready to absorb that 7.3 kilocalories per mole to drive a different reaction. So a reaction that requires energy gets that energy from the hydrolysis of an ATP. And then eventually ATP has to be rebuilt. So the ATP hydrolysis is reversible. ADP can be rejoined with a phosphate group, but again, then you're going to put energy in. All right. Now, how do we do that? That's a long story. But the cell has ATP. Right now, we're not going to worry about where it comes from or how it builds the ATP. But once it's built, it's holding some energy, and then when it's broken into ADP plus PI, inorganic phosphate, that releases some energy that the cell can use to do work. And what kind of work does it need to do? It needs to maintain order. It needs to keep rebuilding things because the universe is trying to move towards breakdown. All right, catalysts. This is not a completely separate part of this lecture, but at first it's probably going to feel like it is. What a catalyst is in chemistry, not just in biology, but in chemistry, beyond just biology, it's a substance that speeds up a reaction by lowering the activation energy. Nothing can violate the law of thermodynamics, so that goes without saying, I would say. All right, it doesn't change really anything about the reaction, it just makes it go faster. And remember, I said that a lot of reactions in the cell would not occur fast enough to be consistent with the cell staying alive unless enzymes are there. And so in cells, the catalysts we're talking about are always going to be enzymes. Outside of biology, there's things you can do to reactions to make them happen, but in biology, they're always enzymes. Enzymes are usually protein. They can be made of RNA, and they can be made of a combination of RNA and protein. I would say the protein ones are the ones we tend to focus on quite a bit. It's the ones we know the most about, to be honest. The discovery of RNA enzymes is much newer, and so we just don't have as much that we know about them. But enzymes are catalysts, biological catalysts that lower the activation energy of a reaction to speed it up. And enzymes are, like, if we're thinking about proteins, remember proteins have a shape. They have a primary, a secondary, a tertiary, and a quaternary structure that gives them a very specific shape. And they typically, one particular kind of enzyme will only catalyze usually one particular reaction. You have thousands and thousands of different reactions that go on in the body in different places, and each of them is catalyzed by a different enzyme. That's amazing. And each of them only interacts with its own correct substrate, which is the reactant. It's amazing. And so when you're studying a reaction, maybe in a different class or in anatomy, if they don't mention the enzyme, there is an enzyme that's there. It's responsible. It's just they don't always tell you what it is. But there's always an enzyme, because enzymes are governing everything in the cell. Because remember, energy flow, transfer and transformation is really the definition of life. The interaction between, the movement of energy into and out of a cell, the movement of matter into and out of a cell is what defines what life is. And so the enzymes, because they are moving energy, they are critically important in life. It is life. So an enzyme has a three-dimensional shape. In this picture, it looks like a circle, but the substrate, which is the reactant, matches the shape of the active site of the enzyme. So the place on the enzyme where the reactant will interact is called the active site. And those two things fit together. The substrate enters the active site, and then the enzyme kind of hugs down on the substrate even tighter, and that pushes and pulls on the substrate and makes the reaction happen. Details for any particular reaction are, vary quite a bit, but that's generally what's going on. Then the products are formed as a result, and the products are released. The one thing I don't like about this drawing is then you go back to the beginning. So then the enzyme is back where it was, and it can go again and again and again. As long as there's substrate available, it will continue to do this reaction. It doesn't get used up. This picture is just trying to show you more of a three-dimensional image The gray globby thing is the protein enzyme. And then this little stick structure is the much smaller molecule that is the substrate. And it fits into the active site. And then, like I said, the enzyme hugs tighter around the substrate, and we call that the induced fit, the hugging. And then that pushes and pulls on the substrate and causes the reaction to happen. We used to say that the substrate fits into the active site like a lock and key. It is kind of like a lock and key, but it's like a lock and key where the key only kind of fits at first, and then the lock kind of reacts and hugs tighter around the key. Lock and key is not a perfect analogy anymore, because I don't know of any lock that will hug the key once you put it in there, like squeeze it. So I don't know. Anyway, that's the model. Now, what's important is that cells need enzymes to do every reaction. Reactions, no matter if they're endergonic or exergonic, they need some energy, whether it's activation energy or whether it's energy to drive an endergonic reaction itself. And all of this is provided by the enzymes. All right. And so, like I said, any reaction that happens in the cell or in the body, there's an enzyme that's involved, and usually more than one. Usually it's more of that metabolic pathway concept where you have enzymes that work in sequence to make a final product. But in any case, enzymes are very compartmentalized. By that we mean that within the cell they are localized to certain places, and a lot of enzymes will need to stay within that compartment. So in the cell there are compartments where there are very strong enzymes, and they do their job, but they only do them within that compartment. If they were released from the compartment, they would destroy the whole cell. It's a little bit analogous, or quite a bit analogous, to like the digestive enzymes in your stomach and small intestine. If you get in a car wreck and you actually get an injury that opens up your stomach or small intestine, if those enzymes get out into your body space, you will start digesting yourself. You will digest yourself. So those are powerful enzymes, and they need to stay in their correct compartments. If they stay in their correct compartments, everything's fine. All right. But enzymes do everything. They do all the energy transfers and transformations, or let's say they're a key component of those energy transfers and transformations. So if a cell doesn't have, if an enzyme isn't working, then that particular reaction won't happen. If you want to kill the whole cell or kill the whole organism, you can disrupt all of its enzymes. So because shape is critical to an enzyme's function, if you boil, so high temperature boiling, or a big change in pH, very acidic or very basic, if you drop a lot something in acid, you will denature it. If you boil something, you will denature it. You will denature the enzyme. You denature all the proteins, not just the enzymes. But the enzymes are a critical part of what you're denaturing. And that's one definition of death. I mean, it's metabolic death. So metabolic death would be where the enzymes in the cell aren't working, or there's no energy transfers and transformations taking place. So those things will work. And I mean, if you think about it, if you boil an egg, or if you heat up an egg on the stove, you denature all of its proteins. But there are other ways to cook something. You can drop it in acid. So I think it's like ceviche, where you can drop, I think it's acid, where you can cook something with just a very acidic solution, not heat. But it gets the same effect. So you're denaturing the protein. So if you denature all the enzymes, then you will effectively prevent them from functioning, and If all the enzymes in a cell are denatured, then the cell will die. All right, so let's, and if there's just a slight shift in pH or temperature, sometimes you can just get some kind of reduction in the function. Might maybe make the enzyme work slower, but if it's more drastic, then you can get a total denaturation. Salt will also impact protein structures because part of the tertiary shape of a protein is formed by ionic bonds, and salt, you remember, is formed by an ionic bond, and so the salt will actually interact and disrupt the ionic bonding in the three-dimensional tertiary level of the protein. Anyway, there's a lot of other things, but these are the fundamentals. So high temperatures, extreme pH, salt can affect all proteins, but specifically we're talking about enzymes. All right, so how are enzymes regulated? Well, a cell can actually either produce enzymes or turn them on or turn them off. There are ways that cells turn them on or turn them off, or they make more or they get rid of them. So they either turn them on or off, or they just get rid of them altogether. So a cell can control what's being made, and then can control whether or not they're functioning or whether they're kind of there's a way to turn a protein or an enzyme off. It's still there, but it's just not doing anything. That's usually a kind of a temporary situation, but enzymes can be regulated. Enzymes can be regulated through what we call inhibition. Inhibition is something that happens in the cell, but this would be, like I said, kind of a temporary way to turn an enzyme off. So it's in some cases it's a natural thing, but some inhibitors are not natural. So things that are poisons, toxins, venoms, actually it's venin, I think. Venin. I don't want to get into that argument, but we'll say venom. A lot of these are enzyme inhibitors. So when you study how a particular toxin or poison or venom acts, usually to kill something, it's because it is an inhibitor for a very important enzyme, typically a respiratory enzyme, something involved with respiration. But in any case, so you have an enzyme. Here they're showing it kind of in this little Pac-Man drawing here. And you have the active site. They're calling it the substrate binding site, but we're calling it the active site. And the substrate fits into that active site with the correct shape, right? And sometimes you have inhibitors that block that site. So here's a picture of an inhibitor, could be a poison that blocks the active site. Sometimes you have an inhibitor that attaches somewhere else, but it causes the shape of the active site to be affected, messed up. Then you can have an inhibitor that here's the one I meant. This inhibitor binds here, and it prevents, I don't know what all these arrows are, frankly. I mean, I know how enzyme inhibition works, and this is a complicated picture. But sometimes the inhibitor binds directly to the active site and prevents the substrate from binding. Sometimes it binds somewhere else, but it has the effect of preventing the enzyme from binding. And I don't think this drawing does such a great, it's not such a great one. They should have had, yeah, I don't understand this picture. This should have been where, when this binds, then the substrate cannot bind. But anyway, all right. So sometimes it's permanent. Sometimes the inhibitor will bind permanently and block the enzyme permanently. Sometimes it's temporary. Usually, naturally occurring enzyme regulators will not be permanent, but the ones that are poisons and toxins tend to be more permanent They tend to bind more permanently to that enzyme. So any inhibitor that binds directly to the active site is called competitive. So that term, I think, is helpful. And then any inhibitor molecule that binds somewhere else in the enzyme and it changes the shape of the active site is called non-competitive. And then any enzyme that, any inhibitor that binds permanently is called irreversible. And any inhibitor that binds but it's temporary, that's called reversible. All right. So I already showed you this, when something binds to a site other than the active site, so like this blue thing is not binding directly to the active site, but it's binding somewhere else, that's called an allosteric site. So you can have, the active site is the actual place where the substrate binds. And then an allosteric site means any other little niche that has some importance. So an inhibitor can bind to the allosteric site, and I like this picture better because then you see how it changes the active site shape, and now the substrate can't bind there anymore. So that would be a non-competitive inhibitor. Now, if there was an inhibitor that bound directly to the active site and blocked the substrate, that would be called competitive. So all this slide is showing is that the binding site where the inhibitor might bind, other than the active site, we would use the term allosteric. All right, there are some cases where a molecule, and I wish they hadn't drawn the square, because the molecule would be a different one if it was an activator, but it can also bind to an allosteric site and actually activate the enzyme. It can go either way. I mean, I'm not saying that one enzyme can do all of these things. I'm saying in some enzymes, there's an allosteric binding of an inhibitor. Other enzymes require an allosteric binding of an activator. So different enzymes have their own particular rules for how they can be regulated and inhibited. And so at each case, each enzyme that you study, if you get that deep into it, you study each one individually. Okay? You can find examples that have all these different features. All right, let's talk about cofactors. Cofactors and coenzymes. These are things that some enzymes have to have in order to function properly. You're probably aware that you need iron. There's a protein in your blood called hemoglobin, but you need iron to make the hemoglobin work. It's not an enzyme, but it's a protein, and it carries oxygen. And so if you don't have the iron, then it doesn't carry oxygen. And so same with enzymes, more specifically, they have usually these metal ions that they need. Magnesium is actually the most common one. Magnesium is the most common cofactor for an enzyme. Now, not all enzymes have a requirement for a cofactor, but 9 times out of 10, if there is a requirement, it's going to be magnesium. Iron is sometimes a required one, zinc, copper. So there are certain ones that you tend to see in different, like I said, different enzymes have their own specific rules for how they have to function, but it's not unusual to have an enzyme that requires one of these. In slang, we would call these minerals, like you take your vitamins and your minerals. Minerals are metal ions. And then coenzymes, I usually tell students to think of coenzymes as vitamins. So you've got your coenzymes and your cofactors. What am I saying? You've got your vitamins and your minerals. All right. Vitamins, though, are not just single atoms. They're actually kind of complicated structures. So here's vitamin B2. It's kind of a complicated structure. So you don't need to memorize the structures or anything. When you take nutrition, or if you've already taken nutrition, you probably recognize these. But vitamins are more complex. They are organic molecules, meaning they have carbon in the backbone. And so vitamins are coenzymes. That's the more scientific term for it. And minerals are cofactors. And some enzymes have to have both. Like some enzymes have to have, a vitamin B and an iron, So the rules for each enzyme are specific to that enzyme. And you're not, you don't have to know any more specifics than that. All right, so a very common thing that happens in a metabolic pathway, and we're kind of circling back to what we talked about at the very beginning of the chapter. Remember, a metabolic pathway is a series of reactions, each one catalyzed by a different enzyme, each one taking the product of the previous reaction and as the substrate for the next reaction, and so on and so forth. And you end up with a final product typically at the end of a metabolic pathway. One thing that tends to be pretty common, it's not uncommon to see, is that sometimes the very end product of a metabolic reaction series, a series of metabolic reactions, sometimes that end product binds to one of the enzymes in the series. In this case, they show enzyme 1. Sometimes the end product can also act as an allosteric inhibitor. Remember, allosteric means something that binds to an enzyme at a place that's not the active site, but it causes the active site to have a change in shape. And an allosteric inhibitor would also be called a non-competitive inhibitor. That means the same thing. So sometimes the end product of a series of reactions acts as the allosteric inhibitor for one of the first enzymes in the series. What does that do? Well, let's say the reactions are going and the end product starts to build up, like the cell doesn't really need that much of it. And so let's say you have a whole bunch of these gold squares and it starts to pile up. What will happen is some of the gold squares will shut down the first enzyme in the pathway, and that will temporarily stop this pathway. But it's temporary. Like I said, things that the cell naturally does are always going to be temporary. It's kind of like a thermostat, right? And once the gold squares start to be used by the cell, and the cell needs to start making more again, then this gold square will release from the allosteric site, and the enzyme will be reactivated. And this whole process is called feedback inhibition, where the end product of a metabolic series becomes an inhibitor for one of the enzymes in the series, but it's a reversible inhibitor. And when the metabolic pathway needs to be turned on again, the allosteric inhibitor will release from the enzyme. And then the enzyme goes back to its original shape and starts working again. It's pretty cool, all right? But these are naturally occurring in the cell. These are not poisons or venoms because they're reversible, and so it's more of a thermostat effect. When in your house, your thermostat, if it gets too warm, it kicks on the AC, but it only kicks it on until the house cools down, and then it shuts down again. If it gets too cold, it kicks on the heater, but it only heats up until it gets warm enough, and then it kicks off again. So in this way, it's a regulator trying to maintain just the right amount of gold squares, not too much, not too little. That's what the cell wants to do. It wants to manage and have just the right amount and not too much or too little. All right, well, there's a lot in these chapters, and you're probably going to want to also look at your textbook as you're going through this chapter, because it's somewhat less tangible than some of the material that's come thus far. So energy transformations, enzymes, it's quite a bit. But that's the end of my lecture.